for the rust, right? So we said that they can be heteroecious or autoecious, so this is a heteroecious form, right? And this has the complete complement of all 5 spore types. And this is Puccinia graminis, which is the wheat rust Gesundheit, which is basically one of the best characterized rusts that we have. So some of these, again, we said that basically it may be that the life cycle is actually heteroecious, but they've just studied it on one plant. And indeed, a lot of these names, so we said there's literally thousands of names in this group, right? It may be that some of them have just been described off of one of the alternative host plants, usually the crop plant, right? Because that's kind of how this works. Someone finds it on a crop, it's described as a new rust species, but no one really has the time to do all the ecology and really trace the full complement of the life cycle. Does that make sense? So a lot of these may be actually heteroecious, but it may be that just the complete life cycle is missing. So the one thing I don't want you to forget about these is that they are actually in the Basidiomycota, right? So you remember when the teliospores do overwinter, right? They have a phase when they come out, they're going to be diploid, and then they'll move into forming the basidia, right? And then that's where you get the haploid spores produced. Okay? So don't forget that the basidium itself, even though it's not what we're used to looking at, right, for the basidium, right? Mostly what we're looking at for the mushroom forms, and that's the Agaricomycotina, based off, remember talking about agaricus, the agaricoid, agaricus basically typifies that group of mushroom forming fungi, right? And so in that group you have what's called the holobasidium, right? The whole basidium. And what that's really referring to is the fact that there's symmetry in that, right? Because all the ones that we looked at are pretty much symmetrical, and they've got 4-spore arrangement, right? Although sometimes that can be 2-spore.
I must be going around.
We're moving into cold and flu season here, guys. Take care of yourself. So this type of basidium that's asymmetrical, and it has this regular septation. Do you remember what the name of this one is? So we had holobasidium for the Agaricomycotina, and for the Puccidiomycotina.
Unhollow?
No. Look at these and see how they're broken up. What's it mean to break something? If something is broken up and distributed, it is fragmented, right? So these are called phragmobasidia, which just means they're septate basidia that are non-symmetrical, right? And we're going to see the phragmobasidia in the rust when we look at them, in the smuts when we look at them. They're going to look even more bizarre in the smuts, actually, okay? And then we'll see them again when we start talking about the Aphilophorales after the rust. Okay? So the holobasidium is the whole basidium that really refers to that being symmetrical, and that's what we see mostly in the Agaricomycotina, apart from the Aphilophorales. And then this type here that has the regular septation is asymmetrical is the phragmobasidium, right? Here again we get the haploid basidiospores. So again, don't forget about this portion. If you're thinking about what are all the 5 different spore types for the rust, don't forget that the basidium is one of them, right? Because you'll spend time remembering all these other types of spores, right? So we have the haploid spores. Those are going to germinate on one of the host plants, right? They'll form these pegs. Do you remember the name of the penetrating peg?
Hostoria.
Hostoria. Very good, Austin. The Haustoria will penetrate into the upper surface, and then you'll have these mating spermagonia, right? So they have the potential to be a plus or minus mating spring, and they will produce here the receptive hyphae as well as the spermatia, right? So they have receptive hyphae and spermatia. We said sometimes they'll even exude sweet nectar, just like a flower would, right? So these are analogous to really flower organs, right? They're going to be on the upper surface of the leaf of the alternative host, right? And then this is signifying, I don't know if you can read it, but plasmogamy. Do you remember what that meant? We saw that when we talked about the Dicaryomycota. That's when the two different hyphal types that are mating compatible fuse into one. And that's when you get what? Dicaryonization, right? Because these guys are going to, again, be N plus N, dikaryotic for a good portion of their life cycle, just like the mushrooms were, okay? So their nuclei will stay segmented, okay? So up here we have this spermagonia with the receptive hyphae and the spermatia. Again, these represent just different mating types. It's not like the spermatia is really male in the sense, it's just a different mating type. And then what happens? Once we have plasmogamy, we have fusion, we have a dicaryonization, and then we have these isha with the isha spores they form. And those are again going to be N plus N, dikaryotic. Now what's the purpose of the isha spores then? To go off and infect the alternative host, right? So you produce a lot of spores. These can be, we said, pigmented. These can be ornamented. So, but the Uridinia spores tend to be more highly pigmented and more highly ornamented, right? But the idea is, again, usually the host plant, the first host is growing around the area where these guys would be growing, so at the edge of the fields, right? The isha spores are released into the field. They infect the alternative plant, okay? Then you have the isha spores are germinating on the wheat. There you start to have infection, right? And then you have on the stem, for example, or on the leaf parts, then you have these Uridinia with Uridinia spores, right? And again, that's awesome. You said this one is for infecting the alternative host. This one is for reinfecting that alternative host, reinfecting itself, essentially.
Since it's like so close to that alternative host, why does it have those like needle-like things to fly further? If it's
because they like think about a wheat field, right? So if let's imagine this building is the wheat field, right? The first alternative host would be like these garbage cans here. They'd be surrounding this wheat field. But the wheat field potentially could be huge, right? It could be the whole size of this building times 10, right? And so if you have a couple of these host plants that are infecting the field, then those get onto a couple plants right here in this position. Then those spores want to blow out over this entire field, right? Same thing, you can imagine like corn, right?
Oh, they're saying it was going to reinfect that host that's already on. So like, why would it need to?
Because this is just one plant. If this is a wheat field, this is one wheat plant in a whole plant of thousands of wheat plants. Does that make sense?
Yep.
So they got to get off there and do the job of going out and infecting that whole field, right? Does that make sense? Because this guy is just going to infect. You know, these host plants don't have to be that abundant. They just have to be there. If they're there, they can get into the crop. And once they're in the crop, that's when, and you saw the rust, how it really destroys the plant. That's because this plant is basically totally dedicated to reinfecting other wheat plants, right? it's been really repurposed by fungus to be an effective agent, right? So you're gonna make a lot of these spread out through the entire field, and by the way, probably onto the next field, right? Does that make sense? But all you have to do is have a small pocket of, say, barberry that's there, ready, right? Have you guys ever seen barberry before?
No.
This is showing the leaves here, but they're not really that super abundant. Okay, so you produced the Uridina with the Uridina spores. Again, this is showing these are N+N, so you have further segment of the dikaryotic station. And then we have the Telia is produced, it can be produced out of the Uridina, and then we have these Telia spores here at the beginning of the winter. They are dikaryotic. When they emerge at the beginning of the spring, they become diploid, and then they form bacilli. Okay. All right. So do you guys get that? So you're going to have to reproduce that life cycle, so study it well. Okay. So after you get all your Halloween candy.
You mean draw it from scratch or just fill in the blanks?
Okay.
I don't have that kind of memory.
Come on, this will be your big challenge. This is a goal. It's good for you, Alice. Okay.
You'd think.
30-point section.
Yeah, this is going to be one of the big questions.
Darn it.
Okay, you guys can do it. So remember, I mean, there's some little tips that you can do, right? You know these are in the Basidiomycota. So what's one of the spore stages? Basidiospore, right? Then what comes first, the Aeschyspore or the Uridina spores? Aeschyspore, because A comes before U. That's what I said, right? A, and then U, and then just remember that the Teliospore is the overwintering spore. And then if you know these are Eschyl spores, you know that they come out of the Eschym, right? If they're Uridina spores, they come out of the Uridinium. Easy. And the nuclear stage should be known pretty easily too, because it's really just like the mushroom again, right? Here it's haploid, and then as soon as it starts forming the real rust spores, then those are all basically dikaryotic until you get to the formation of the basidiospore, right? Which was exactly the same pretty much as the mushroom. Okay? So you can do it, Alice. Okay, so we talked about these macrocyclic. This just means that all 5 spore states are present because we know them again. Okay? So that's the state that we just saw. It had basidiospores, Aeschyspores, Uridina spores, and then it had the Telia spores, right?
If you get rid of that Uridina cycle, like how much would the spread decrease? Is it like a huge percentage?
I mean, it could be potentially, right? So we went through all of these. Remember that you can produce this sweet sticky. So think of this. So the spermatia and the receptive hyphae are part of this phase, right? They're part of the 5th type of spore that you have. Okay? So they are haploid, and again, they're analogous to kind of like the having plant parts, right? Having the pollen is the male fertilizing element, right? So the spermatia is kind of like the pollen. The receptive hyphae is kind of like the ovary, essentially, stigma style ovary, although there's no real ovary structure. Okay. So here's the spermagonia again. They do have the orange color. We talked through about all these, right? And I think we were.
I think we ended at the last.
Right. Here. Okay. So here are these teliospores. The teliospores have been really important for the taxonomy, right? So again, if we look here, Teliospores, Proboscidean, okay. This is a dormancy requirement, but the Teliospores provide the basis for taxonomy. So looking at the structure and gesundheit again of these Teliospores has been important for describing the different species, right? Okay. And so here's the Teliospores breaking out of the telium. And then here you see kind of these characteristic forms when you look at them under the microscope, right? So here's a two-celled Teliospore, and they're darkly pigmented, right? So you can see this is dark, kind of grayish orange again. Okay. So the spores are orange in these rusts, and they, although they can verge on being blackish, although we'll see the smuts will be black, and then this is characteristic of taxonomy.
They almost look flagellated.
Right, because they're basically, do you see this right here? So they're basically what? A piece of hyphae that inflates and gets a thick wall around it. Right? So this is actually like a hyphopeg that they're going to be growing off of.
Okay.
And then these are just showing they're broken. So I don't know if that was in nature of them falling off or somebody pulled them out of the telium. Does that make sense though?
Yeah.
I mean, these are all really a form of what's called a chlamydia spore, which is just a spore that forms on the end of the hyphae. There's no specialized basidium or anything other than the telium. Okay. So here, this is showing various types. You notice that some of them they're showing with that hyphopeg there. Okay. And here type C is Puccinia that we were just looking at. Right? So the two, and we had to correct that in the homework, right? Anna, that was one of the ones that, so yes, indeed, Puccinia is characterized by the two-celled telium. Right. So we have these other types. So here's Eurymyces, that was just a single spore. So they just used to look at these and they would find the plant that they were on and they would say, okay, it's got a single cell telium, it's on this plant, we'll call it Eurymyces matus or something, right? But notice there's some interesting ones. This F, for example, Phragmidium, so phragmo, again referring to the broken up nature, the septate nature of the spore, right? And this one, Cenodochus, is very interesting, gesundheit. And then finally H, Ravenelli here, that's very interesting. Right? So there is some variation, and remember this is a huge group. And the last thing I want to point out here is notice that these are by J.C. Arthur, 1934, Purdue Research Foundation, Lafayette, Indiana. So Arthur, remember when we met Stephen when he was here? Stephen works down at the herbarium or the fungarium down there, and it's called the Arthur Herbarium. So there's the Arthur, which is the rust collection, and the Kriebel, which is plants and some other fungi, right? So this guy was a researcher that was really known for studying these rusts, right? And Kathy Aim, who's Stephen's mentor, has now picked that up.
I have to know that name for the test.
No, but I just wanted to point that out because, I mean, this is part of our Purdue heritage, really, which is kind of interesting. I'll get some slides on him at some point because that'd be, I like to look into the history of these people. I do know that when he left Purdue, that he took a large chunk of his collection with him because he had invested some of his own personal money in traveling and collecting these rusts, right? So he took his collections home, and the dean told him, hey, wait, those all belong to the university. You can't take those home. And so the dean called the police on him and had the police come to his home and try to take the cabinets and the collections away, because apparently they'd taken them out like in the middle of the night or something. And he said, no way, you're not taking this away. But then they when he died, they agreed to leave him to the university, because he basically just wanted to work on them, right?
After calling the police, I wouldn't have given it to him.
I know. Well, welcome to university politics, right? Anyway, so here are those Basidia and Basidia spore, right? So here's that telium. So here you actually see it. This is really interesting. Here's Puccinia, right? The 2-cell, and here is some cytoplasm here, and you notice that the Basidium has popped right out of that teliospore, right? And there you have the fragnal Basidia showing one Basidia spore still left here. Okay. Now what's interesting, the other thing I want you guys to think about is that these guys are a basal group to the Basidia mycota, right? And remember that there was an ancestor that branched, and half of the group went over to be things like mushrooms, right? That have the hollow Basidia, and the other half had turned into the ascomycota, right? So if you look at this, it's kind of interesting to think that you have these Basidia spores laid out